I'm giving this presentation in replacement of Dr. Jorgen Richt, who asked me to send his apology. He's on business trip now. And a little bit about myself. I completed my DVM and PhD at Moscow when I was working as postdoc at Iowa State University in Paul's lab, where I was fortunate enough to do some work, early work on PRRS and porcine circovirus, when I had about 10-year experience of working with industry at Fort Dodge Animal Health and Novartis Animal Health. Again, it was companion animal vaccines and also swine vaccines, including influenza and circovirus. And last 6 years, I'm working at Kansas State University at Center of Excellence for Emerging and Zoonotic Animal Diseases, where Dr. Richt is director and I'm science project manager. So my presentation will be about some work which we are doing on development of new approaches to influenza vaccines. Here there is a brief outline of my presentation. I'm going to give you a brief update about influenza virus and its evolution, when I'll give you a brief introduction to universal influenza vaccine concept, and the rest of my presentation will be about our next concept using vectored vaccines for avian influenza. So this portion of presentation is mostly will be focused on avian influenza. So influenza virus is RNA virus with family Orthomyxoviridae. There are several types of species of influenza virus, and our major focus on influenza A, which is spread widely in birds, swine, and humans. Genome is represented by 11 genes incorporated into 8 segments of RNA. Influenza virus evolved through 2 major mechanisms, which probably everybody know, but I'll briefly repeat. One is antigenic drift, where point mutation accumulates during virus replication, and this change gradually phenotype and antigenic property of virus. And another mechanism due to segmented nature of a virus genome is antigenic shift, when during co-infection, viruses exchange segments, and as a result, a totally new antigenic variant of influenza is emerged. And if this new emerged variant doesn't have, is pretty much novel to environment, and it goes into pretty much naive population, and it spreads really fast among susceptible species, which cause pandemics. And here is an example of evolution of latest pandemic virus, pandemic H5N2, which took place in 2009. This triple reassortant virus containing genes from swine, avian, and human species, and circulating in pigs, reassorted with introduced Asian isolates, and this created pandemic virus. Pandemic H5N2, which was moved to human, and currently this virus is prevalent in pig population and in humans. So the problem of influenza is that virus is fastly evolving very fast, and new antigenic variants emerge. So and there are no cross protection between, or there is only limited protection against different variants of influenza, even within the same, sometimes even the same within the same serotype, as it is a case in H1N1 viruses in pig population. So here is a concept of universal influenza vaccine was developed. And in order to explain you this concept, I need to give some background information on hemagglutinin protein of influenza. Hemagglutinin protein consists of two domains: globular head, which is highly variable and determine antigenic properties of isolate, and more conserved domain called Stalk domain. Globular head is responsible for attachment of virus, and Stalk domain is responsible for virus fusion with cell membrane. And this Stalk domain is, as I mentioned, it's conserved and immunosubdominant. But at the same time, there are some epitopes which are present at this domain. And these epitopes, in theory, they provide broader cross-protection against different variants of influenza. And here, again, there is an example. So concept of universal influenza vaccine is based on shifting of protective immune response toward conservative cross-protective epitopes. Here, if to create several hybrid proteins which have globular head from different serotypes, and for example, H1N1, H5, and H6, like it's shown here, and Stalk domain from pandemic H1N1 in this example. When sequential vaccination with these proteins leads to accumulation of antibodies against this conservative Stalk domain, and in theory, these antibodies should provide increased protection against different variants and maybe against different serotypes. And at the same time, a response to globular portion provide additional protection against different serotypes. So this is kind of universal. This is universal vaccine concept, and several studies were done in mostly in human medicine to test this concept in ferrets and mice and in non-human primate. In our lab, we also tested this concept, and we used 3 hybrid proteins. One, all of them have globular Stalk domain from pandemic H1N1 virus, and globular domain against H9, H8, and H5 serotypes. So prime vaccination was with H9, H9N1, or H9H1 hybrid protein. Secondary vaccination, first boost was with H1, and third one was with H5N1. All booster vaccination and challenge were done at 4 weeks apart, and unfortunately, in this study, we did only one study on this. All piglets enrolled in the study had maternal antibodies to pandemic H1N1. We always use piglets for our work and from the same farm, and we use it for several years, but in this case, when we acquire piglets, it happened that we have H1N1 outbreak. But nevertheless, here are all these piglets were challenged with pandemic H1N1, and we got some promising results. Our virus titer in nasal swabs of universal vaccine groups was decreased on 5 DPC. It's 2 logs versus 5 logs in controls. Virus was not detected on bronchial alveolar fluids, and also we observed significant reduction in macroscopic lung lesions. this was our first study, but our data were similar to that, generated for testing of human vaccines in ferrets, mice, and non-human primates. So this concludes my first part of my presentation about a universal vaccine concept, and we are planning, if funds will be available, we are planning to further develop this concept. And the rest of my presentation will be on another approach. We are trying to use vectored vaccine, vector-based vaccine as vaccine for influenza. And here our choice is Newcastle disease vector. Newcastle disease virus is family Paramyxoviridae. It's single-stranded negative sense RNA genome, approximately 15 kb in length, and reverse genetic system for this virus available, and it allows to manipulate with virus genome. And to create, and we are using NDV attenuated Lasota strain, which is safe in poultry, and million doses are used almost every day. So we don't worry about safety of this vaccine constructs. So in order to create vaccine candidate, HA gene is inserted between P and M genes of Newcastle disease virus, and then after rescue, we have vaccine candidate ready for trials. And this whole process, here we work with our collaborators in Mount Sinai School of Medicine, and they develop this technology, and it takes them from 2 to 4 weeks to develop this vaccine candidate. So here are some points on advantages of NDV-based vaccine platform, because this for using poultry. We're able to differentiate infected from vaccinated animals, which is important for control of outbreak in case of introduction of foreign strains. System developed is allow to develop new vaccine candidates very fast, so within 4 to 6 weeks from sequencing of HA gene of new isolate to production of vaccine candidate. And it's also in poultry, it prevents both NDV and influenza infection. And we are fortunate enough here to work with our industrial partner, Avimex, who has a license for this technology, which is acquired from our collaborators in Mount Sinai School of Medicine. Recently, we had a chance to test this technology during H5N1 or H5NX outbreak. As you all remember, last year in the United States there was a big outbreak of avian influenza virus, and this outbreak was caused by Eurasian H5N8 isolate, which was first detected in December 2014, and then virus recombined with low pathogenic avian influenza strains circulating, reassorted with low pathogenic avian influenza, resulting in appearance of H5N2 variant, and then another variant which was detected during this outbreak, H5N1. Overall, 219 outbreaks were detected, and as countermeasures, 48 million birds were slaughtered, 40 million chicken and 8 million turkeys. And overall economic loss is estimated as $4 billion. And what's interesting, original isolate, H5N8, was detected only in three cases. Majority of outbreak, 216 out of 219, were caused by certain H5N2. And H5N1 isolate was detected only in wild birds, and it never spread to domestic poultry. So as soon as these strains become available by USDA, we acquired them, and we did some study to compare pathogenicity of all these 3 strains. Here we enrolled 6 groups of chickens in the study, and 3 groups we challenged with high dose of each of these strains, and 3 groups we infected with low dose. And in high dose, all chickens die within 2 to 5 days. And when we use low dose, we notice some difference. None of chickens die at 10 to 3 logs, TCID 50. None of chickens died in H5N1 infected groups. About 50% of chickens died in H5N8, and 70% of chickens died at H5N2. So this result correlates with what happened in the field, where H5N2 caused the most damage during this outbreak, and this virus we selected for our vaccine trial. At the same time, while we were doing this study, our colleagues at Mount Sinai School of Medicine, they developed a vaccine strain. They put HA gene from Iowa isolate of H5N2 from this outbreak. They confirm expression of HA gamma-glutenin by IFA, and they send it to us for trial. And here is design of our experiment in chicken. We enrolled 3 groups. First group was vaccinated with 5 to 10 to 6 PFU of live virus. Second group were 5 to 10 to 8 PFU of NDV-H5 killed virus. And third group was vaccinated with placebo. Vaccinations were 2 vaccinations, 2 weeks apart, and challenge with virulent H5N2. Turkey isolate from Minnesota from the same outbreak was done 2 weeks post-second vaccination. And here a pretty much major slide which show result of this trial. All our vaccinated groups, all chickens survived, and all chickens in control group died by day 4 post-infection. This is just a photo showing these chickens on day 4 post-infection. And we also didn't observe any viral shedding in swabs, only some probably leftover virus from challenge, initial challenge or on day 1 post-challenge. And also no virus was observed in tissues. When we share this data with our colleagues at Avimex, we were very excited, and they asked us to do one more study to evaluate mass application of H5 NDV-H5 vaccine, where we use coarse-spray vaccination. So we never done this study before, so we just purchased a household sprayer from Walmart and put chickens in a box. We play a little bit with sprayer, and pretty much on day 0 we spray chickens with approximately 10 to 6 TCID 50 per dose, and 20 days post-challenge, post-vaccination, we challenge them with the same H5N2 virus. And here there is outcome of this trial. This time we didn't get 100% protection, but 70% of our vaccinated survived until the end of post-observation period, 14 DPC, exhibiting only minor clinical signs, except for those 3 chickens which died. And all control died within 3 days post-challenge. So still for us it was encouraging result because we never did this before, and we didn't use commercial sprayers, and dose was pretty low. And here also we observed some virus in vaccinates, with some shedding in swabs, but if you see here 2 out of 11, it's kind of correlated with mortality for our study. So probably some chicken didn't acquire enough virus to vaccine virus to ensure them protection. So I think we can improve this technique significantly. So in summary and conclusions, universal vaccine, influenza vaccine for pigs seems feasible even in the presence of maternal antibodies. At this point, vaccination scheme with multiple vaccination rounds make universal vaccine not feasible for swine industry, but technology develops very fast, and we still want to pursue this approach. And at minimum, we can use this as a swine model, as a model for human vaccine trials. But our pathogenicity studies show that different virulence of isolates of H5N2 of different isolates emerged during a recent outbreak. And both our vaccines, live NDV-H5 vaccines, live and killed, they completely protect chickens from virulent challenge. Mass application of live NDV-H5 vaccine for chickens also seems feasible. And to summarize, this NDV-H5-based live and killed vaccine are promising vaccine candidates in chickens against intercontinental H5N2 viruses, and is suitable for mass application. We did some studies for this NDV-HA-based vaccine in pigs, and we get some promising results, but unfortunately I cannot tell more due to some IP issues which we still have to address. Thank you for your attention. And last but very important slide, acknowledgements. It's Dr. Richt and Dr. Venjuma groups at Kansas State University, ICA group, Adolfo Garcia Sastre at ICANN School of Medicine, working on NDV concept, and also a group of Florian Kramer, who is at the same School of Medicine, which is not on this slide. We're working on pursuing Stalk vaccine. And I would like to thank our sponsors, and also my thanks to CMG Group at College of Veterinary Medicine and people at BRI for great assistance with animal studies. Thank you.